Phases of gastric secretion. Gastric secretion is said to occur in three phases, that is 1. Cephalic phase, 2. Gastric phase, 3. Intestinal phase, as shown in the figure 67.6 and table 67.1. Right, now let's have a discussion over this. You can see in this figure how the cephalic stage, how the gastric phase, how the intestinal phase are there in the gastric secretion process. Now let's talk about the phase and its description and its regulation. Now now let's discuss about the different phases and the description and the regulation of each phase. Right, cephalic phase. It occurs even before food enters the stomach, especially while it is being eaten. It results from the sight, smell, thought, or taste of food, and the greater the appetite, the more the intense is the stimulation. This phase of secretion normally accounts for about 30% of the gastric secretion associated with eating a meal. Note this point. What are the stimulus for gastric secretion in the cephalic phases? 1. The sight, smell, thought, or taste of food. These were and the appetite were the stimulus for gastric secretion in the cephalic phase. Important point. How do you regulate this cephalic phase? Neurogenic signals that cause the cephalic phase originate in the cerebral cortex and in the appetite centers of the amygdala and hypothalamus. They are transmitted through the dorsal motor nuclei of the vagus and thence through the vagus nerves to the stomach. This is how the cephalic phase regulation occurs. How the neural signals originate in the cerebral cortex and in the appetite centers of the amygdala and the hypothalamus. Then they are transmitted through the dorsal motor nuclei of the vagus and finally via the vagus nerve to the stomach and stop the gastric secretion. Right, now let's discuss about the gastric phase. Gastric phase occurs once food enters the stomach, which in turn causes the secretion of the gastric juice during several hours while food remains in the stomach. The gastric phase of secretion accounts for about 60% of the total gastric secretion associated with eating a meal. Regulation. This phase is regulated by 1. Long vagovagal reflexes from the stomach to the brain and back to the stomach. 2. Local enteric reflexes. 3. The gastrointestinal mechanism. This is how the gastric phase is regulated. Now let's talk about the intestinal phase. When food enters the upper portion of the small intestine, say probably duodenum, particularly in the duodenum, small amounts of gastric juice continue to be secreted. This phase accounts for about 10% of the acid response to a meal, right? Response to a meal. How it is regulated? This phase is regulated by nervous mechanisms too, hormonal mechanisms, probably partly because of small amounts of gastrin released by the duodenal mucosa. Gastrin is released by G cells, as we know, secreted by G cells is secreted by G cells secrete the gastrin. This is a brief overview of the phases of gastric secretion. You can see how the cephalic phase is regulated. See, now coming to it is it is occurs when there is a smell, sight, thought, or taste of food, or even appetite stimulates this cephalic phase. See how it does. Cephalic phase in this the neurogenic signals that cause the cephalic phase originate in the cerebral cortex, right, and in the appetite center of the amygdala and hypothalamus, right. Now they are transmitted through the dorsal motor nuclei of the vagi and thence through the vagus nerve to the stomach. See here cephalic phase via the vagus. Parasympathetics excite pepsin and acid production, as we know. You can see the signals are coming from the vagal center of the medulla, then through the afferent fibers, then they relay in the vagus trunk, then they get dispersed into secretory fiber and local nerve plexus, right. Now let's look at the gastric phase, how it is regulated. It is regulated by local nervous secretory reflexes, 2 vagal reflexes, 3 gastrin histamine stimulation, right. We can see now how the intestinal phase is regulated. The nervous mechanisms, 1, 2, hormonal mechanisms. Now let's talk about the inhibition of gastric secretion by other post-stomach intestinal factors. Although intestinal chyme slightly stimulates the gastric secretion, say it accounts for about 10% of the acid response to a meal, right. Intestinal phase, gastric secretion. Although the intestinal chyme slightly stimulates gastric secretion during the early intestinal phase of the stomach secretion, it paradoxically inhibits the gastric secretion at other times. Important. It inhibits the gastric secretion at other times. This inhibition results from at least 2 influences. 1. The presence of food in the small intestine initiates a reverse enterogastric reflex transmitted through the mesenteric nervous system and intrinsic sympathetic and vagus nerves that inhibits stomach secretion. These reflexes can be initiated by distending the small bowel by, by the presence of the acid in the upper intestine, by the presence of protein breakdown products, or by the irritation of the mucosa. That this part of the complex mechanism discussed in chapter 74 for slowing stomach emptying when the intestines are already filled. Please remember these. Now the reverse enterogastric reflex. What are the stimulus? One is distension of the small bowel. 2. The presence of acid in the upper intestine. 3. Presence of protein breakdown products in the upper intestine. 4. Irritation of the mucosa. These were the stimulus for the, that is, enterogastric reflex, reverse gastroenteric reflexes. Right. Now 2. What is the second influence? 2. The presence of acid, fat, protein breakdown products, hyperosmotic or hypoosmotic fluids, or any irritating factor in the upper small intestine causes release of several intestinal hormones. One of these is secretin, which is especially important for control of pancreatic secretion. However, secretin opposes stomach secretion. 3 other hormones, say gastric inhibitory peptide, which is a glucose dependent insulinotropic peptide, vasoactive intestinal polypeptide, and somatostatin, also have significant to moderate effects in inhibiting gastric secretion. The functional purposes of intestinal factors that inhibit gastric secretion is presumably to slow passage of chyme from the stomach when the small intestine is already filled or already overactive. In fact, the enterogastric inhibitory reflexes plus inhibitory hormones, say gastrin and usually also reduce the stomach motility at the same time that they reduce gastric secretion, as discussed in chapter 74. Now let's talk about gastric secretion during the interdigestive period. The stomach secretes a few milliliters of gastric juice each hour during the interdigestive period, that is, when little or no digestion is occurring anywhere in the gut. The secretion that does occur is usually almost entirely of the non-oxyntic type, composed mainly of the mucus but little pepsin and almost no acid. Unfortunately, please remember the secretion during the interdigestive gastric secretion during the interdigestive period. It is of non-oxyntic type, that is composed mainly of mucus but little pepsin, almost no acid. Unfortunately, emotional stimuli frequently increase the interdigestive gastric secretion, which is highly peptic and acidic, to 50 milliliters or more per hour, in which in much the same way that the cephalic phase of the gastric secretion exerts secretion at the onset of a meal. This increase of secretion in response to emotional stimuli, say emotional means your sight, your smell, your thought, especially thought, will increases the gastric secretion, Emotional stimuli is believed to be one of the causative factors in the development of the peptic ulcers, as discussed in chapter 75. Right now, let's talk about the secretion and activation of the pepsinogen. Several slightly different types of pepsinogen are secreted by the peptic and mucous cells of the gastric glands. Even so, all the pepsinogens perform the same functions, right? When the pepsinogen is first secreted, it has no digestive activity. However, as soon as it comes in contact with the hydrochloric acid, it is activated to form the active pepsin. In this process, the pepsinogen molecule, having a molecular weight of about 42,500, is split to form a pepsin molecule having a molecular weight of about 35,000. Pepsin functions as an active proteolytic enzyme in a highly acidic medium. Say optimum pH is 1.8 to 3.5, but above a pH of about 5 it has almost no proteolytic activity and becomes completely inactivated in a short time. Hydrochloric acid is as necessary as pepsin for the protein digestion in the stomach, as discussed above. As hydrochloric acid is necessary. Now let's talk about the secretion of the intrinsic factor by the parietal cells. Now the substance intrinsic factor, essential for the absorption of vitamin B12 in the ileum, is secreted by the parietal cells along with the secretion of the hydrochloric acid. When the acid producing parietal cells of the stomach are destroyed, which frequently occurs in the chronic gastritis, the person develops not only achlorhydria, but that is lack of stomach acid secretion, but often also pernicious anemia because of a failure of maturation of red blood cells in the absence of vitamin B12 stimulation of the bone marrow. Right?